Hello, this is Oliver Fien. I am very happy to welcome Dr. Evi Mathé. Dr. Mathé joined the NIH National Center for Advancing Translational Sciences in 2020. So there she is heading that division, and before she was at Ohio State in bioinformatics as an assistant professor. Before that, she was again at NIH as a postdoctoral student. And today she will talk about enhancing our understanding of metabolomic and multi-omic data through robust data analysis workflows. So I know Evi Mathé because she is also a member of our MANA, Metabolomics Association of North America board. She is very active in the SODA working group, which is software and data exchange. So we have met many times, and I'm very happy to meet her. Actually, I also met her in Canada the other day, and I learned a lot just by sitting next to her, and when she told me about a lot of platforms that I did not know. So I hope that we will see more about that today and can learn together. Dr. Mathé, the floor is yours.
All right. Thank you, Oliver. So let me share my screen. Yeah, can you see?
Yeah, I can see it.
Okay, perfect. So before I dive into kind of the efforts we've made in metabolomic and multi-omic data, I want to tell you a little bit about translational science at Ncats. Ncats is a fairly new center at the NIH, and the overall mission is to catalyze the generation of innovative methods and technologies that are going to enhance development, testing, and implementation of diagnostics and therapies. And so basically the mission is really to provide more treatments more quickly to all people. And our IFX team is in the intramural portion of Ncats, and our mission is to produce these data-driven decisions and to accelerate translation. And we do this by developing new methods or enhancing the application of existing methods. And I want to point you to this figure on the right here. This comes from a publication, an Ncats publication from our previous director, Chris Dawson, in 2019. And it kind of shows the characteristics of a translational scientist. And they're very broad, right? So it involves being a systems thinker, a skilled communicator, domain expert, process innovator, boundary crosser, team player, rigorous researcher. That's a lot of things. And when I first saw this, I got pretty excited about it because I think a lot of those characteristics are present in folks that are doing informatics or bioinformatics, because we are by nature, I think, very collaborative, because informatics is pretty much used everywhere. And even in the sense of multi-omics, there's a lot of know-how and a lot of different types of knowledge that are required to pull on these projects, particularly large-scale projects. And so this particular figure spoke to me both in terms of understanding translational science, but also thinking of it from the informatics and multi-omics point of view. So maybe some of you see some of yourselves or parts of yourselves in this as well. Okay, so diving now into data and multi-omic data specifically. So the amounts of data are getting larger and larger. The complexity is also increasing. And of course, that means that there's more analysis need. So if you look at this very short version of a timeline here, before the 1950s, all of the measurements were mainly qualitative. And then there was a switch to quantitation in the 50s to the 80s with different technological revolutions, HILIC, for example, or some of the earlier sequencing days. And so that increased the capacity to be able to measure more analytes, right? More genes, more metabolites in a single sample. And then in the 90s, 2000s, and beyond, these technologies became more accessible, right? So cost was reduced, and also they were able to be implemented more in-house. And so now, not only do we get the depth in terms of the coverage and amounts of analytes we can measure, but it's being applied to much larger sample sets and much more complex study designs than what was possible in the past. And to put this into a little bit more concrete context, I want to point you to this work that was published by the Gerstein Lab about 10 years ago. And this figure is relative to sequencing data. So I generalize this, I guess, to multi-omics. And so I want to refer to the colors in this plot with the legend here on the right. So anything that's in red is effort that is related to study design and biospecimen collection. Anything in blue are efforts that are related to omic measurements and generating the data. And anything in gray is about data management. So how do we store the data? How do we capture, and what meta information do we capture pre-processing? And then finally in orange here, this is about the stats and also the ability to interpret the data. And so with that legend in mind, if you look at around 2000, the bulk of the effort on the y-axis here, we have percent effort. The bulk of the effort was on data generation. Very little effort in study design because the studies weren't as complex. Little bit of effort on data management, maybe a little bit more on the stats. About 10 years later, which is when this particular paper was published, you can see that there's an increase in the percent effort spent on study design, also an increase in the stats, a lot less effort spent on generating the data, more effort spent on data management. And their prediction, which I think is pretty spot on, was that now there's even more effort on study design, even more effort on the stats and network analysis, and relatively little effort on data management and generating the data. And what I really want to emphasize here is the importance of involving informatics, bioinformatics in the study design and biospecimen collection to be sure that you can actually do the stats and the interpretation from the data that you want. And I think this is going to do nothing but increase in terms of the effort in study design. So with all of this complexity, one way to look at it is you have all these different omics that you can put together. I've listed some here. Obviously, this is not exhaustive. And typically, these molecular measurements, they have to be viewed in the context of other factors. And for humans type studies, those can be understanding exposures, maybe demographics or lifestyle factors, or other phenotypes. And there has to be this metadata that's associated with these molecular measurements for any of it to make sense. And then all of this information, so the clinical demographic type information and the molecular information gets analyzed, and there's two various approaches, and at a broad view, there's two broad approaches to combining this data. One is to actually look at the relationship between the different omic features. And this may be looking at correlation type relationships, either globally or phenotype specific, for example. But then typically you get a list and you want to see whether the list is meaningful, right? Are those relationships meaningful? Are the single metabolites, genes, et cetera, are those meaningful in themselves? What is known about them? And for this, researchers are going to rely on a priori knowledge, right? So one, of course, can go to PubMed and go through a bunch of papers by hand to understand their system. But there's also a lot of curated sources that are out there, or community-based sources, or even in silico resources, or a mixture of all of those that are out there and that can be leveraged. And really, we've worked in both those areas over the years. And then, of course, at the end of the day, you want to be able to interpret the multi-omic data in the context of biology, chemistry, and disease. And so this is going to involve, again, those methodologies on predicted relationships. I'm giving examples here on the right, and pulling in the information. So, you could look for new relationships, you could make better predictions, you could apply some network-based analyses and look at differences between networks, looking for novel mechanisms. There's all sorts of applications. So I want to point you towards IntLim, and we have this work under review right now, and we've made some enhancements that I'd like to share here. And the goal with this is to capture phenotype-specific associations. And so if you look here on the top left, you have an example of gene abundances on the x-axis, and then metabolite abundance on the y-axis. And if you look for an association across all the samples, you do not see any trend. However, if you separate the samples by the different types, in this case, there's a red group, say a cancer phenotype, and a blue group, say a non-cancer phenotype. And now there's a pattern that emerges. And here there's a positive correlation, association between these two in the red group, but a negative association in the blue group. And this is what we're after, because this is one way to discover potential, maybe physical interactions between analyte types, or they could be indicative of these strong associations that are very characteristics of whatever phenotype you're looking at. So we capture these systematically with a linear modeling approach that has an interaction term. Conceptually, the interaction term looks at the difference in slope between these lines, and it will pick up these types of interactions. Now, we've recently generalized the code so that you don't need to only look at genes and metabolites. You could look at, say, metabolites, or genes, or other types of omic data that you may have. And one can also adjust for covariables. And these covariables may be adjustments of technical artifacts like batches, or clinically relevant, or study design relevant covariable adjustments for human, such as age, BMI, et cetera. We also can now support continuous phenotypes. So of course, we can't show this as easily as it's shown here in this figure with a binary phenotype, but we can support continuous phenotypes. And because IntLim actually evaluates every single possible pair, right, between two lists of analytes, sometimes there's a huge amount of models that are run. It can get into the 1 million plus. And so we've performed some cross-validation and null permutation models so that we can assess the robustness of the results and further filter down into results that are less likely to be artifactual. So we do have in our package, and also have a server access that basically doesn't provide any programming in order to use. I will say that right now it is down because we've had some difficulties in our systems, but it should be back up very quickly. Well, something happened. Oh, there we go. All right. So building on to IntLim, we've been using IntLim for different, is there a question in the chat? I'm okay with folks asking questions as we go.
We'll hold off for questions at the end.
At the end, okay, sure thing. Okay, so I want to highlight this particular application where we did leverage IntLim, and this is looking for pathways and general omic features that are relevant to lung function. And we had 2 independent asthma cohorts. This is a collaboration with Drs. Jessica Lasky-Sue and Rachel Kelly at Harvard. And these 2 cohorts had metabolite measurements that were very different between both cohorts. And so if you looked at specific metabolites that were in common between both, it was very hard to find because there wasn't that much in common to start off with. And so I have Tara, who's a PhD student who has put together these networks where you have nodes that are metabolites or genes, and you have edges that represent whether there's this phenotype specific association between the 2. And you'll note that anything in purple are analytes that are in common between both, and anything in green or in orange are nodes that are specific to each cohort. But what's interesting is that when we look at these networks and start performing pathway enrichment, we found 2 pathways that are very much related to asthma. And so, of course, we have the insulin resistance, which is associated with asthma. And then there's also known asthma therapies that are targeting GCPR. So, so we're excited about this approach, and you know, this is unpublished, so still a work in progress. Another application is our goal of finding analytes that are relevant to drug response. And for this, we're using the NCI 60 cancer cell line data, which has metabolomic and gene expression data. And this is a similar type of network where you have nodes that are genes and metabolites, and you have edges that correspond to a relationship that is phenotype specific between 2 nodes. And you can analyze this network. Of course, you know, networks have all sorts of metrics that can be applied onto them to find regions of most interest. Here, we looked at Betweenness Centrality, which is a measure of how much traffic a node can get. And if you look at the top ones, the ones that had an identified metabolite, there was definite support in terms of the involvement of that metabolite and drug response. So again, this is unpublished, and you know, we're trying to generalize a little bit this method, but here you can see how the approach was applied in 2 different contexts. So I want to switch gears a little bit now and talk about inferring meaning. So this is, again, less about looking at relationships between the different omics, but more about trying to infer meaning out of them. So your input is typically some type of list of relevant analytes, maybe how much they are associated with a phenotype of interest, maybe also what their relationships are in terms of the numerical data. And, you know, some basic questions that one may ask are, what types of analytes are they? So for metabolites, that may be looking at chemical classes or chemical structure. For genes, that may be knowing whether they're non-coding or coding. For proteins, that may be whether they're enzymes or DNA binding proteins, et cetera. Typically, researchers also want to know what types of biological functions they may have. And so with this is looking at biochemical or regulatory pathways. And then finally, we often want to look at these lists in terms of, have they been found in other diseases or associated with other diseases or similar phenotypes? So there are many resources that are out there to ask these questions, and each resource typically has a slightly different slant, whether the focus is more on diseases or the focus is more on the metabolites, or it's a conglomeration, but then maybe there's not as many annotation types, particularly for the metabolites. And so we were trying to develop ways to infer meaning and found that a lot of methods were actually not using the same resources. And it was really hard to compare across the different resources and compare across the different pathway enrichment tools. And so for better or for worse, a couple years ago, we started building RampDB, and this is a relational database of metabolomics pathways. So it is focused on metabolites. It also does contain annotations for genes and proteins, and it does so from multiple sources. So here you can see broadly the types of annotations that it holds. It has information on pathways, on properties. So these chemical properties, for example, are structural properties like SMILES or INCHI or INCHI keys. We have information on chemical classes. So different lipid types from lipid maps, for example. We also have the ontologies pulled in. These ontologies are from HMDB. And you know, we're working with them to pull those. And so those may be involvement in certain diseases or biospecimen location, those types of ontologies. And finally, we're pulling information on reactions. And this is basically to group together metabolites and genes that are involved in the same reaction. And it is not denoted in this figure, but our development branch right now of this work includes pulling reactions from RHEA, so that we can expand on this and explore more network analysis methods there. So when we built this, we also wanted to be sure that it was open source and standalone, so that anybody could use it or you could incorporate it in a different tool. And this incorporation in a different tool is important because, again, the idea was to create a source that is comprehensive, that is pulling many different types of information from different places, and that can be used for method development. We're able to sustain this because we've developed a semi-automated ID mapping, which is helping to limit the amount of curation that has to be done, which is very time intensive and also quite expensive. And we do have access to RAMP as an R package, or we also have its contents that are accessible through a web API, and we also have a user-friendly web interface. So this really can be used for computational and non-computational users alike. So some recent improvements that I would like to highlight, we have expanded the content, and I'll show that in a minute. We're including a lot more in terms of lipid annotations and also in terms of chemical structures and class information. I've already alluded to the semi-automated anti-resolution scripts that we came up with. And we have additional functions now in the R package to interact with the database. So chemical class enrichment is a new one. Now for the Fisher's pathway analysis, one can select their own background. And we've also worked on our visualizations for reporting the pathway enrichment results. And finally, we do have a new front end that got completely redone, and the public facing API is proving to be quite useful. So for you to get an idea of the coverage, we have over 250,000 distinct metabolites, over 15,000 genes and enzymes, and over 50,000 pathways that are represented. And in terms of the new chemical descriptions, we have chemical properties for over 250,000 metabolites, and over class information, chemical class info on more than 181,000 metabolites, and ontologies on over 150,000. So with all of this information in one place, we can do just some high-level analytics. And this figure in particular is trying to understand how many metabolites are mappable to biological pathways. And they are grouped into their chemical classes. And so you can see that there's certain chemical classes, for example, nucleosides and nucleotides and analogs that have a quite high representation, 23%, compared to some others, for example, mixed metal, non-metal compounds. And this type of visualization is pretty useful because if you have a new metabolomic data set, you can use RAMP and very quickly get a sense of what biological space am I actually covering with my particular data set? So I want to talk a little bit about the entity resolution and how it's being done in a background. And so again, RAM pulls from multiple sources, right? And each resource it's going to have its own linkages between metabolites. So in this example, in resource 1, metabolite 1 has ID ABC that all refer to the same metabolite. Metabolite 2 has IDs D and D that refer to metabolite 2, and then there's IDF here that refers to metabolite 3. Now in the second resource, you see the same pattern for metabolite 1, IDs ABC, same pattern for metabolite 2, but now look, metabolite 3. Metabolite 3 is linking IDF and IDC. So when the data gets harmonized, C, because it's in common between metabolite 1 and metabolite 3, they are getting pooled together, and we're assuming that it's metabolite 1. But what happens if one of these is wrong? Like in this case, what happens if this particular linkage, CNF, is wrong? The harmonized naming is going to be wrong. So we found specific examples of this. One example here is this wrong association between this particular molecule with this molecule, which are clearly very, very different, but were erroneously linked. So we tried to figure out how we could detect those in a semi-automated way, because for a long time, those were detected in an ad hoc way, just based on data we were looking at, or based on users that would tell us, hey, this is wrongly linked. And we put together a very simple test looking at mass differences. So just comparing the molecular weights between all possible IDs and making sure that they were not beyond a certain threshold. And if they were flagged as a potential wrong mapping, and those were manually curated. But then they're output into a new ID mapping file that gets read in so that it's those, once they're curated once, they're automatically fixed. All of those are publicly available if that's useful for other efforts. And we found in some instances some fairly large discrepancies in terms of the masses between molecules that were mapped together based on the individual resources. And so you can see the distribution here. And so we're very open to hearing about if there's any thoughts about other metrics that could be implemented to help with this and to help flag some of these mismappings, which we know exist. Going back to ways of interacting with RAMP, I want to highlight some features that enhance the usability of RAMP. So one feature is that one can input mixed IDs for metabolites, genes, and proteins. And this is particularly handy, especially for metabolites, which I'm sure most of you, if not all, are aware. It's really hard sometimes to have data sets that have just a single ID type for all metabolites. All you have to do is prepend the ID type to the metabolite ID, and then off you go. I will say that there is an option to input names. We are still trying to optimize this. Using names personally scares me. I don't think there's so many synonyms and so many issues there that we try to push users to use IDs. So other features include the ability to run single or batch queries. So you can either input, a single analyte or multiple analytes and look up what pathways they map to, common reactions, ontologies, and chemical properties. And then we also have pathway enrichment and class enrichment, and there is clustering of the enrichment results that happens. And this is important because remember, we're pulling information from multiple resources. Therefore, there's going to be redundancies in some pathways. Some pathways, meaning some pathways are going to appear in multiple sources. So they're going to appear multiple times in your results. And so, you know, we put together a clustering algorithm, which is based on the Jacquard distance. So it's the similarity of pathways based on how many of their entities within the pathways overlap. Through a series of merging, you can cluster the pathways. And here's an example result here, where the cluster 1, for example, is grouping together sulfur amino acid metabolism pathways coming from different resources, or sometimes it's grouping together similar pathways within a single resource. So this just helps navigate the results a little bit more easily. Another expanded functionality is the ability to define a background. So one can either use all analytes in RAMP, or one can use a particular ontology. So for example, if you have a plasma data set, then you can narrow down your list of background to plasma metabolites, or you could input your own user background. And the chemical class enrichment is something new. So I want to talk a little bit about the importance of appropriately defining your background. This is a simulation that we ran on randomizing real metabolite data 500 times. And so because we are randomizing the data, we do not expect any positive pathways or enriched pathways to result. And so we ran this analysis using 2 types of Fisher's test. One using a custom background, which is shown in orange here, and another one using a KEGG background, which is often used. And what we found is that when you're using a non-specific background, in this case KEGG, you get a lot more false positives than when you do not. And however, it's all of a balance also with false positives, and I'll get back to this a little bit later, but paying attention to the background that you're using is definitely important. This is just a picture of the front page and the different types of analyses that one can do, and also a snapshot of the API access, which is documented and available to you through the Swagger page. Okay, so a little bit more on these broad analytics. What one can do also is understand the overlap of the coverage in metabolites and genes between all the different resources. And this is focusing on metabolites and genes that map to pathways. And so here you can see these are upset plots, and each row is a single database resource, KEGG, HMDB, Reactome, and Wiki pathways. And then each column, each row, each bar plot here is showing the number of metabolites that correspond to whatever has a solid dot here. So in this case, there are 817 that are unique to Wiki pathways. There are 286 that are unique to Reactome, etc. And so globally, there's only 786 metabolites that are found in all 4. So I think this really shows, and it's similar with the genes, where there's 4,600 genes that are found in our 3 sources for the gene annotations. And so clearly, combining information from these different resources is going to expand the coverage and the annotations that you have. We're also looking at ways of looking better into the overlap of pathways. Ncats has actually come up with some algorithms, and they do rely on quite a bit of curation to aggregate pathways from different resources. This effort is called Bioplanet. And so, we're exploring a little bit if there's ways of doing this potentially with RAMP to just simplify a little bit the information content. And so we've just started plotting some metrics here. And here we're plotting max Jacquard distance. So, Jacquard again is a similarity that's based on the number of common genes between two lists A and B divided by all the analytes in A plus B. And what we find here, so what we find here is on the x-axis, we have that max Jacquard index. And I'm just pointing out some things here because we were trying to understand how similar pathways, how this metric did it, particularly when you have sets A and B that are of very different sizes. And so I want to point you to this. 0.8 has a, Jacquard is from 0 to 1, first of all. So the closer to 1, the more similar. And 0.8 is pretty high, and it's very clear that these are two pathways related to apoptosis. Now, here, we also have pathways that are very related. They're dealing with surfactant metabolism, but their Jacquard index is only 0.35. And that's a little bit artificial because one of them is much smaller than the other. So we're exploring different metrics to try to deal with this and figure out exactly what we want to put here in the denominator to look for these similarities. Another aspect that we can look at is promiscuity. So we define promiscuity by the number of pathways in which a gene or metabolite participates. So oftentimes in applications, we want to find analytes and genes, in particular in translational research, that eventually we can target. And if you target a metabolite or gene that's involved in a lot of different pathways, that means that at a system level, it's going to affect a lot of things. And so it's more likely to have some type of adverse events. And so here we're just looking at how many pathways metabolites are present in. On the left here, you can see the distribution, and then the right, you can see the distribution of the genes. And there's definitely quite a bit of metabolites and genes that are highly promiscuous. And so we want to be able to use this information to try to narrow down our efforts of what we would look more into and further validate or try to further target. Okay, so I want to talk about metabospan, which we've worked on this for quite some time, and this was an attempt to ameliorate Fisher's test. Fisher's test is still very, very extensively used in multi-omics and metabolomic and multi-omic analyses. And we thought that we would use RAMP to try to see if we could improve a little bit some of the enrichment methods. So overall, the method works like this. There's a network that is built based on RAMP. And again, the nodes here are metabolites and or genes, and the edges are some type of relationship between them. This relationship could be based on a shared pathway activity, or it could be based on chemical similarity for metabolites, or what other functional annotations you may want. Now, using this as a background, in a sense, the user then inputs two sets of data. The first set of data is the metabolites of interest. So these are your altered metabolites. And then there's also a reference, which is the reference background that is input. So these may be the metabolites that you're measuring, or you can use a generic background. And what the algorithm does is that it maps these metabolites of interest onto the RAMP knowledge graph, and it expands the list based on metabolites that are nearby on the network. And it does the same for the reference panel. So, in a way, you're expanding the list of the metabolites of interest and the reference panel based on what you know about these metabolites from RAMP. And the reason for doing this is, again, a lot of times with metabolomic data, you don't get totally complete coverage. And so there's a lot of data that's missing, maybe because you're not picking it up, or it's not in the sample altogether. And so after we've expanded our list of metabolites of interest and reference panel, then we perform pathway enrichment analysis. So the results are very similar to pathway enrichment analysis. And these are some metrics that we put together for analyzing the performance. And if you look up here on the top right, this is a false positive experiment. So again, we're shuffling real data and we're not expecting to see any positives here. And so you can see the different methods that are tested. I've highlighted in red here the 2 methods that do best. And those are the Fisher's method using a custom background, and also Metabospan using a custom background. Other methods have much higher false positive counts. NetGSA is also, I would say, pretty low. Now, if you flip this on its head and you look for true positive data. So here we took about 75, we randomly selected pathways, and then took out 75% of those metabolites in the pathways. And those were used as our input list of altered metabolites. And then we asked the question, can we retrieve those? And definitely here, metabolite set enrichment analysis does a far superior job than any other method. And Metabospan is a bit in the middle. And so, you know, we're trying to understand this a little bit more and tease this out. You know, what's the best background in what context? When do we care more about true positives versus false positives? And how can we understand this a little bit better, just from a usability perspective, to also understand what is the most ideal method to use for interpreting these metabolomic and multi-omic data. So, you know, some uses in future developments. Again, there's many ways you can interact with RampDB, and we're also always open to incorporating new information. And so, you know, if there's interest in different annotation types that are not present right now that you would find useful, just contact us. And we're also incorporating this in several other projects. I've listed some here, including Translator, which is an Ncats project that also aggregates many different types of knowledge sources and tries to create an interface to be able to do very basic queries on all of these resources with minimal user input. Metaboanalyst, I think I have to describe to this group what that is, and also Comets Analytics. So I want to talk a little bit more about Comets Analytics because I think it goes back to how do we conduct robust analyses, and how do we standardize our analysis pipelines, and also the resources we use and the processes we use. So Comets is a consortium of metabolomic studies. It is NCI-led, and it's really about creating a community of large cohorts to facilitate conversations and the ability to perform large-scale meta-analyses. And so all the Comets cohorts have blood metabolomics data that's available in at least 100 samples. Now, for Comets Analytics, the goal for creating it was to streamline consortium-based meta-analyses of metabolomics data. And we did this by standardizing data, models, and the results. So some of the design features that we considered are to eliminate the need for data use permissions and consent. So each cohort can run Comets Analytics on their own cohort and then send their data to whoever is performing the meta-analyses. We also wanted to minimize the burden on analysts to harmonize data and run models without errors. So there is a component here for harmonizing metabolite names, and the running models without errors. There's a lot of automation that happened, which is, you know, at the back end, a lot of logic that was incorporated to automatically detect when there are covariables that are too strongly correlated, so one should be removed, or covariables that have too many missing values, and those are removed, or sometimes a model can't run because that cohort just doesn't have enough samples to run that particular model. And all of this is done in an automated way so that there's no errors that are returned, but there's a log that's returned that tells you what happened. And we also have a user-friendly access for those that do not code. The four basic parts are, again, standardizing the input, automating the integrity check of the input data and the harmonization, automating the model validity checks. And then this fourth step is what we're currently working on. And that's the, well, actually, I'm sorry, I misspoke. We do have the results that are standardized, but their aggregation is currently what we're working on. So performing the actual meta-analysis. So a little bit on the features here. There's a generalized process for running models. So now it's super simple. It's literally 3 functions at the minimum, one for loading the data, one for defining the models, and one for running the models. There's many different types of models supported. I have them listed here: partial correlations, GLMs, logistic regression, proportional hazard regression. I want to point out the extensive testing that we did. So we tested this 2 large-scale analyses that included 45 cohorts. So this comprised 68 data sets with over 4,500 metabolites measured in over 130,000 individuals. And this spanned 15 different platforms. So it's a pretty substantial amount of data. And we tested the robustness of Comets Analytics on 2 main criteria. The first one was, how easy is it to prepare the data input? I think one of the challenges with any tool is just to figure out how to parse your data so that it can be appropriately input into your software. And there were minor issues encountered, but all of them were resolved by a quick email to one of us that were developing Comets Analytics. So it wasn't a huge issue to get over that hump. And then in this process, we've also made our documentation more clear. The second criteria was to correct the processing of the input files. So this deals with the model checking. And we were able to run all of these models without any errors. And so that is quite a big feat, and we were pretty pleased with being able to do this, and it provides a lot of flexibility in using Comets Analytics. So in this next release, we're expanding the ability to harmonize metabolite annotations across different platforms. We're working on an automated process right now to complement the curation. I do not have any slides on this, but happy to discuss. As I mentioned, we're doing the meta-analysis, and we're also incorporating RAMP for chemical and pathway enrichment analysis. I do want to mention as well that while Comets Analytics was designed for cohort, large-scale meta-analyses, it doesn't have to be, right? Because in terms of translation, for example, if you have data on murine models or on cell lines or on humans, and you're trying to look for commonalities across these different data sets, you could certainly use Comets Analytics for that purpose as well. So I think there's much broader usage that's possible here than cohort level analyses. Oliver, you already mentioned SODA. I'll make a little plug for SODA in case there's folks that don't know about it. So SODA is a software and data exchange, which is part of MANA. And we do have upcoming SODA meets. And the format of these is that we have typically 2 individuals that give a talk either on their data that they generated and they think would be useful to be for benchmarking, or their tool that they're developing or have newly developed, and they're trying to get users or get some input in terms of making it better. So this is really about coming together as a community to get info on how current software data and data analysis results are being used. So this really should help everyone, and I think it's a way to also bridge the gap between computational and non-computational users. Okay, So I know Oliver and I had talked about this and how there's so many different tools out there, and which ones to use, and why are we still developing tools, and I'll answer that last one and say that there's no one size fits all, right? So there's a lot of algorithms that exist. Many are going to require slot modification to answer your specific question. And so there's still a lot of development to be done. It also depends on what question you want to answer. And the devil's in the details there, right? Because depending on what your input is and what you want to answer, some methods are going to be more or less appropriate to use. And I think that as a community in the computational world, I think we need to do a better job at clearly delineating the usage, and it can be beyond, beyond its initial implementation purpose. Of course, there's also lots of aspects in terms of, the validation. How will you validate your hypothesis, and how can this information actually go back to the tools, right? Are there certain tools applied in certain contexts that, are doing a great job at validating? There's always a lot of utilities that need to be built internally to help bridge information. This is going back to the parsing, right? We're struggling right now, actually, in terms of disease annotations, because a lot of disease annotations in some broader databases do not capture rare diseases well. Rare disease is a big focus of Ncats, and so we want to be able to use these common resources for rare diseases as well. But, diagnoses of these is very difficult. So ontologies for rare diseases is something that is very, very difficult. So we're having to build a lot of internal utilities to be able to bridge these types of gaps. Also, standards are always evolving. And that means that there's new connections to be made. And we're always going to have to be able to deal with new data types. And this is very, very nuanced, right? This ends up getting really into the weeds that most users of these resources do not really understand. And it's always a balance between providing too much information that will go over the user's heads and not providing enough information so that the tools are getting misused. And currently there's a lot of standard workflow pipelines, and many different groups end up building their own, and they're not widely adopted. Like the basic steps, and here I'm thinking, some of the pre-processing type software, a lot of those, the steps are very similar, but they're done slightly differently, from group to group. Does it matter? Probably, because the results are going to change. Does it matter at the end in terms of the interpretation? I think, I think that kind of remains to be seen. So, to help with some of this and to help with creating these robust analytics for multi-omic data, I think we need to be very aware about standardizing and versioning our data. Providing all the details that you all know about in terms of data acquisition, reporting different types of annotations. I think there have been improvements in this. When we started working on RAMP, actually, the different resources were not as interlinked as they are. Now there's a lot more cross-referencing that's going on, and that cross-referencing is super important. So even if different standards are used, as long as there's cross-referencing between the standards, that's critical. We also need to keep breaking down silos and make sure there's some type of consistent data reporting across the different fields. That is not happening. There are multi-omic efforts, but they tend to be disease-specific. And then you have omic-specific efforts that are not disease-specific, but there's no non-specific omics efforts and non-specific disease efforts, at least that I have seen at a large scale, and that's ready to go and publicly usable. So, you know, creating teams that help navigate these different options, that cover different breadths of domain knowledge, and that help with translation and communicating across fields is super important. I mean, this communication and translation, it's not always easy. It's not always easy to translate a chemical task to a software engineering task, for example. I think with that, I want to thank particularly John, Tara, Kyle, Haley, Andy, and Tim. They've a lot of the work that I showed involves one or more of them. Of course, the team is a lot bigger. We're a team of about 34 individuals, and you can kind of check out what we're doing here if you'd like. And so with that, I will stop here and see about questions.
Great.
Yes,
Thanks.
thank you so much, Abby. That was enlightening, very pathway focused, as usual. Our academic coordinator, Dr. Aparna Vinaya, will take it from here and guide us through the Q&A. And please, everyone, if you haven't done so far, please post some questions.
Great. Thanks, Oliver. Thanks, Evie. That was such a great talk. I really like that slide of like how to interpret you know, multi-omics data, and like you broke it down into very simple, approachable like questions. And so I appreciate that. I'll definitely use that in like my teaching. So yeah, starting off, so you had talked about phenotypes and all of this stuff, and I think it's in TLIM, right?
Yeah.
When you have thousands of metabolites and many phenotypes, you will get a lot of spurious false positive associations in TLIM. How do you correct for that?
Yeah, so we correct for that two ways. One way from a numerical perspective. So we do a lot of permutation testing to be sure that the pairs that are significant wouldn't show up in null data. And then we also do cross validations where we make sure that the significant pairs are showing up in a sufficient number of the cross validations that we do. And the cross validation, I'll caveat, sometimes it's a little bit difficult if you have small sample sizes. So I will caveat there. So that's one part. In terms of the interpretation part, sometimes we have the knowledge that we can use, right? So if we're looking at relationships between genes and metabolites, that's when we may look at reactions, like how many of those are supported by reactions, or how many reactions away are they from each other, right? To try to get rid of some of these spurious findings. If we're looking at metabolite microbe data, there's this really nice resource, and I'm gonna say the name wrong. I think it's NJC 19. Am I right? Oh, sorry, I can't remember. I'll email the group, but it's they fairly recently came up with an update where they're mining PubMed to look for microbial metabolites that are either taken up by microbe or produced by microbes. And so then we've used that database to see, well, how many of our IntLim, you know, pair results actually supported, you know, by this information. So we do try to do kind of high level sanity checks when we're there, but I still think that there's probably going to be false positives there, and you know, that's for you to figure out what you want to pursue as a user, I think.
Yeah, of course. And a follow-up question to that: associations can be done by GLM or Spearman rank and other ways. How many samples would you need, and which type of technical measurement errors to solidify associate true positive associations and distinguish them from false positives? So really, how many samples at the end of the day do you need?
Yeah, so we've tried this on really small sample sizes and also on larger ones. And obviously, the smaller your sample size, the worse off you're going to be. In a general way, so when we do regression models, there's this rule, which, you know, to be perfectly honest, is as arbitrary as the p-value of 0.05 rule, but where you know, if you have, say, like 10 samples, you shouldn't have more than 2 covariables in your model, right? So typically 5 samples per covariable used in your model. And so then it depends how you build your model and what you're going to adjust for. Clearly, with correlation analyses, it's interesting. We actually did some simulation analyses. This was a long time ago. This was probably like 7-8 years ago, where we looked at the effect of correlation metrics as the sample size decreased. And what happened is that your distribution became more skewed. So you either got really close to 1 or really close to 0 and less in the middle. You do not see that as much with the regression models. I will point out, though, that obviously regression models are not reversible. So it depends what you have as your outcome. Correlation metrics are. So, you know, there are some tweaks to understand there on the back end, but, you know, we do try to make that clear, you know, for the users in NITLM. So, yeah, I mean, you can always, you know, you can apply any of these tools on small data sets, but, you know, I mean, if you're looking at 1 million associations and your sample size is 10, then I wouldn't read too much into it, to be frank.
Gotcha. Thank you. On the drug response, in the cancer cell unpublished data, you added functions to metabolites from literature and pointing to Metabolon data. Which functions or databases do you use to comprehensively associate functions to metabolites, here specifically with cancer metabolism? And we, you know, Oliver had asked this question. He thought the answer was RampDB, but the next question says maybe not. So
yeah, I'm seeing this now. That's funny.
Yeah.
For this particular figure, she actually went and hunted down papers one by one. RAMP is going to have your basic biological pathways. So when we looked for overall biological pathways represented, we do use RAMP for that. This was kind of a very specific question as, you know, as it related to the impact of the drug treatments. And so we don't have any annotation that is geared specific to that right now in RAMP.
Okay. So on this topic of RAMP, do you also cover cells or like other tissue, like specific tissues, or at least like enrichment, for example, sphingomyelins that are involved in myelin sheath in the brain? And like what extent of other aspects does RAMP cover?
So a lot of these annotations are present in RAMP. We currently do not have the ability to do enrichment on those because we're trying to figure out how to put it together in a meaningful way, right? So do you end up doing enrichment on, you know, each individual type of annotation, or is there a way to, you know, combine them and look at it a little bit more holistically? And honestly, we haven't really figured that out. So I'd be curious to hear thoughts on this and what would be, you know, more useful. Sometimes I find it difficult to look at enrichment results that have a conglomeration of different types of annotations because I get my wires crossed, but maybe, you know, maybe that's just me. So I guess I'll spit that one back out at you and hear, you know, hear your thoughts on this.
Oliver?
Oh, Jesus.
It was your question.
I guess. No, I know. I had so many questions, and Evie knows that. So when you go to protein databases, there's, a lot, or gene databases, they always have funky names and functions, like activate so-and-so, and right? And for many other, like both proteins and genes, you would also know, like, oh, this is typically, enriched in so-and-so tissue, or, so you would have functions from that side. Whereas in metabolites, and also in RampDB, you always go like, oh, this is citric acid, it's in the TCA cycle, and it's important in the mitochondria, and yada, yada. Kind of, it's, I don't want to say missing the point, but it's just focused too much on how to make things rather than the physiology of things, right? So the physiology would be, sphingomyelin has one, if I'm a brain researcher, I have a mouse brain as a data set, then I want, and maybe I'm not very smart, maybe I'm a chemist. Oh, sorry, I shouldn't say that. I am a chemist. But maybe I have never heard about sphingomyelins and myelin sheaths, right? So how do I get this kind of information out when I'm not a domain expert? In this case of the brain. And once you are a domain expert, that's fine, but many chemists, or metabolomicists, today they work on fish, and tomorrow they work on mouse poop, and then the next day they work on whatever, right? So they cannot possibly know all that.
And I think a lot of the ontologies that we're pulling through HMDB are going to help with this, because they have some physiological information. They also have some sometimes manufacturing, environmental type information, and so it's just that right now we're still trying to understand the coverage and the breadth of this type of information and how we can use it. But that's a very good point, and you're absolutely right that a typical, chemist or informatician is going to see a whole bunch of different contexts and a whole bunch of study designs, and it's really hard to communicate out meaningful results without having that domain knowledge. So point taken about seeing how that can be extracted a little bit more clearly, using something like RAM.
Ideally, you would like to have something like a response from your tool, just like you showed in this little graph on the cancer cells, right? Where each of those metabolites had a function that was related to, cancer cells. And your function was not like oleic acid is a fatty acid, which is, it has a double bond and is, made by, desatyrases, ta-da, right? Which is, would be also true information, but not relevant in this case. So,
Great. If there isn't any more on that, I'd like to continue with some more questions. I had a specific question on this slide where you showed the errors of incorrect linkages. What percentage of incorrect, what was the percentage of like the incorrect linkages or like the mismapping? And then how is that being corrected now, especially because like there are so many other databases involved in RampDB and you're pulling information from those databases?
So I cannot remember off the top of my head. Let me see if I can link to it real quick while I answer the second part. I will say that it's not a huge amount. So that's the good news. So it doesn't kill the whole thing. The way we deal with it is basically we track it. So whenever we see mismappings, we actually tell the resources that there is a mismapping, right? So that they can go back and potentially correct them. But sometimes that takes time, right? Because it takes time and resources to correct those things. And so then what we end up doing is fixing them, but in a way that we can kind of automate that process. And so we've seen if we've curated something before and we've seen it, then we don't have to recurate it again, and we just fix it. And if that, so we found 640 to give you a scale. So it's a small
Yeah.
amount. And now there might be some that we're missing, but this is what we found, you know, with our algorithm. And so if that mismapping is found again, it'll automatically get fixed based on our hand curated fix, right? And if it hasn't been found, then it'll just skip it.
Okay.
So
I
it's
see.
pretty versatile, you know, in that way.
Yeah, I mean, it's nice that like you've addressed it. It's small, and you know, there's an automation process in terms of like correcting that.
Yeah.
On the slide with upset plots, it's kind of funny. We just talked about upset plots like last week, so I a little prepared to see, you know, understand what was on the screen there. You had a little message that said like you access the KEGG IDs through HMDB and not directly through KEGG. Is there a reason why? Like, why wouldn't you just go directly to KEGG?
KEGG licenses. So we wouldn't be the new KEGG license. We wouldn't be able to provide that publicly.
I see.
So we have an internal KEGG license, but we can't publicly give that, which is, I mean, that's just kind of the state of things right now. So that's why that disclaimer is in there.
Yeah, that makes it a little challenging, right? With those nuances in terms of licensing and what's available. Yeah, okay.
That's right. That's right.
That makes sense.
And I'm so glad. I love upset plots. I mean, we've been using them for a while now, for like probably 7-8 years, and it's just absolutely brilliant. So
yeah, we had a little debate about that last
really?
week. So, you know, whether to go upset plots or Venn diagrams, right?
Oh, you don't want to go Venn. I'm very biased here. Go the upset way.
But yeah, I liked how the data was presented. It's a nice, clear and, you know, clear way to understand the information.
Good.
Now switching gears to Metabospan. Why do you use Fisher test instead of Kolmogorov Smirnov? That's a hard one to say. Enrichment tests that are not dependent on background databases.
Yeah, and we absolutely could, you know, use these type of self-contained tests. We haven't, because originally Fisher's was so commonly used. And so, you know, we were just trying to kind of build up on them. I think that, you know, the KS tests are used more frequently. And I know that this group, you know, uses them quite a bit. And so we absolutely could. Yeah. And it would, I mean, I think this background business is definitely a big issue. So, yeah.
Okay. Regarding the pathway comparisons via Jacquard distance metrics, if we were interested in considering pathways identified via different functional analysis methods, how are different pathway names handled? Are these manually matched and confirmed, or are they semi-automated approaches?
So RAMP does not try to harmonize pathway names. And it does not necessarily, like, we don't have anything internally. Actually, that's not totally true. We do have a pre-calculated matrix of Jacquard overlap between all the pathways that we have versus all the pathways. But we don't necessarily use that information beyond the clustering. So it's really only after the enrichment that then we try to cluster based on how similar the entities within each pathways are. So they're not manually matched. This is all automated. I am not convinced that Jacquard distance is the best way. And, you know, as I alluded to, if you have differences, large differences in the sizes of two pathways that you're comparing, then you're going to get misrepresentative, I would say, Jacquard distances, either, you know, too large or too small. So I hope I'm answering your question. If not, you know, let me know.
Okay. Nothing in the chat from the person who had asked that question. So we'll give them a second, but we'll move on to the next question. What about biopan lipid pathways?
Yeah.
Have you included these?
We're looking into it. Yeah.
Okay,
Yeah.
awesome. And then on one of the last questions in the chat here, on the slide with analysis to analyze promiscuity, which tools did you use to find the relationships between metabolite and gene? Could you provide some details about this pipeline?
So let me just pull it up to make sure I'm understanding the question. So are you referring to, let me get myself shared again. Are you referring to this plot looking at the promiscuity?
Yes, that.
Yes.
Okay. So the tool that we used is basically RAMP with a script added, and I think that script is probably available because any figure we generate for a paper, we have the scripts there. If it's not and you need it, just contact me. But basically we map to RAMP and we just count the number of pathways that that particular gene or metabolite maps to. So that's what we do. So I'm not sure in terms of how much more detail you're looking for in terms of the pipeline there.
Yeah, I was curious because I have a set of 108 enzymes, and if I would want to look in a specific organism and how many pathway is gene is related, I was wondering how I could use that as an input for the tool to get a count of either metabolites or reactions they are involved.
So you're trying to, so first of all, I will say that RAMP is human specific. So if you're looking at, across different organisms, this is not the right tool. But we haven't incorporated promiscuity as an input to, enhance pathway enrichment. But what you could do is you could certainly look for enriched pathways. And then, you get for each pathway that is enriched, you get the metabolites that are in that pathway. So then for these metabolites in the pathway, you could look at their promiscuity score, right? Their promiscuity, and just focus more on pathways that have, less promiscuities and are more specific.
Okay, thank you.
One last question here in the chat, and it's a good one. Is an association of a metabolite to a pathway enough? That's a big question. I think one of the slides you had mentioned, validation is that the metabolite pathway function, or are we kicking the can down to the biochemist to validate the metabolite function and mechanism by classical biochemistry experiments, creating a new bottleneck in the future?
Yeah, I mean, I think that bottleneck is already there, right? Because you cannot validate every single association. And so you end up going after, associations that are going to be of greater interest. And what you see generally is that, there's huge biases, right? I mean, there's, and it's kind of interesting because there's just certain themes that become popular and non-popular, right, over time. And so these, you're just seeing a lot more coverage in certain areas than the other. I think at the end of the day, if you want to get down to a mechanism of action and you want to drill down to that level, you have to go back to, the biochemical experiments that you're alluding to. Whether that needs to be done at a large scale is less clear to me, because it just doesn't sound feasible, at least with the tools that we have. So then, you end up relying on in silico predictions, and back to the network of RAMP that we had that we've created for Metabospan, that's also kind of interesting to use in itself, because if you have new metabolites of interest or something like that, you can look at what's nearby, right? And you can, these kind of guilty by association type network analyses that you could potentially apply. So yeah, I mean, these associations to pathway, in essence, they're a guide. I think some pathways are going to be targetable, some pathways absolutely not, right? I mean, when you have pathways that get returned, like metabolism is my favorite one, yes, thank you, right? So we actually remove those, like we remove the ones that are too large because they're just completely nonsensical. But, sometimes just understanding, wait, are they really specific? And, you're going to have to drill down into orthogonal pieces of information, including some of the ontologies that Oliver was referring to, in his previous question. So, I mean, point taken, this is, and a lot of studies, that's where you end, right? It's like, here are the pathways that are modified, end.
Yeah, so again, like, I did ask for tissues, but it's also organelles or cell types. Yeah. So with genes and proteins, we often have we already have the atlases and whatnot, right?
Yeah.
They said, like the human proteome atlas, and you name it, right? Also, so many things. We don't have anything like that in metabolism, really. Yeah.
But I think there's a fairly good idea of what the metabolites are measured in terms of at least some biospecimen types, blood, urine, certain tissues. It's fairly broad, but it's there. What we definitely don't have, the annotations are present, and we haven't used them because I'm not entirely sure how reliable they are, is, the intracellular annotations.
Yeah, the organelles, like BMP lipids are only lysosomes, right?
Yeah.
So if that is not an outcome, people would not know about it.
Yeah.
And I think that is really important. The more we dig into metabolism, so the further away we get from classic building and burning pathways like cake, the better our specificity of information will be.
Yeah.
I was intrigued by, you just said, just the funny word targetable. So is this whole thing governed by drug targets like cancer?
No, ramp?
No, I mean, because you said like that, it was like,
in general,
like just 2 minutes ago, like, oh yeah, you have the pathways and then you look for things that are targetable. Yeah. And it's like, oh, okay, that's one type of questions that people do in biology, like cancer or whatever, right? Gonna find drug targets. But that's not always the question in metabolomics.
It's not always the question. No, absolutely. And that's one that comes to mind off the top of my head always, because I mean, those are more of the applications. But I think that does bring up a good point, is that there are so many different types of applications for metabolomics, and that issue that you're raising about having these more specific resources versus not, and I think this is why there's so many resources that are available, and those that are specific to, I don't know, even exposomics or I don't know, transformations of metabolites and things of that sort. I think that information eventually needs to get included, but it's always the hard thing of, you put a lot of information together and then it just gets so much that you can't figure out what you actually need for your purpose.
I have so many more questions. If Aparna lets me.
We should have another meeting.
I know.
Just for
but
this.
Hey, so you also at some point you said you don't, you want as input database identifiers, right? Mm-hmm. So different database from CAS numbers to whatever. What about RefMet? RefMet is an approach to give a unified name to metabolites.
Yeah.
Would that be an identifier you would accept?
Yeah, so we do not accept RefMet currently as an input. However, the algorithm that I mentioned that we're trying to automate for metabolite name harmonization as part of Comets Analytics, we are relying on RefMet. So we could certainly have those as input. The thing is, it gets a little bit tricky, right? And it depends what you want to do, because if you're doing biological pathway analysis, there's a lot of chemical IDs or names that are linked to pathways that are not specific to a single molecule, just based on structural resolution. They're going to be a more generalized structure, right? However, if you're doing chemical class enrichment, or if you're doing, some other types of ontologies, you actually want it to be a single molecule. And so depending on the application, I think is going to depend what type of input would be accepted versus not. And we're trying to work that through. So we do have we are looking into RefMet, and I think it is a great effort. I think it's incomplete, but it's increasing. And they recently put out APIs as well, so it's been a lot easier to work with them as well.
So with that, there's ever more compounds, and RefMet is a human curation effort. Yeah. And you also said you don't want to go into the question of synonyms and whatnot, but somehow we need an automated tool that utilizes names and maps it automatically, and not by some dude in San Diego, right?
Yeah. Yeah.
But it's not going to be you.
Well, I mean, I don't think it's fully automatable.
Okay.
So I think it's semi-automatable. I think the automatable is going to get you to a certain extent. I mean, we are hoping to provide a solution that's good enough. Is it going to be an end-all solution? I honestly doubt it. I don't think you can get around no curation with this type of stuff.
All right.
I may be wrong there, but.
So that's all the questions I had.
Okay, Oliver, thank you. We are a little over time, and I'd like to share my screen to talk about our next speaker. So Evie, thank you so much for giving the lecture and really talking about multiomics, because that's like the next big challenge, and that's where we're going to pull in so much and learn so much more from all the
thanks for all the
omics.
questions. I appreciate all the discussion. Thank you.
Great. So our next meeting is in November 2nd. Dr. Evelyn Rampler is professor in the chemistry department at University of Vienna. She'll be talking about lipidomics and glycolipidomics, but her lab generally focuses on these topics, as well as developing novel LC-MS-based omic assays. In terms of her lipidomics assays, she's really targeting like the communication of lipids with tumor microenvironments. So if you're interested in these things, please join us next month for this talk by Dr. Evelyn Rampler. So thank you all for joining. Thank you for the questions. And again, hope to see you guys all next month.